All right, looks like everyone's in, so let's get started. Hello, everyone. Thank you, as always, for coming to our Funk Biogeography seminar series. Our speaker this month is going to be Carlos Daniel Cadena from the University of the Andes. But before we get to that, I just have a couple of society-related announcements. So first off, you might remember from a couple of months ago, we put out a call for workshops with the small tweak that for clarity, we're referring to them as training courses now, but it's going to be the same solicitation. That said, we have a couple that have come in already. So the society is currently in the process of processing that, figuring out logistics. So keep an eye out for those, and details on course content and registration will come out as they are available. But also, I want to say that the deadline for submission is still through the end of the month. So you have until the end of August to do that. So if you're still interested, this is a reminder. Tips will give up to 1,000 US dollars to the organizers of these training courses. If you're interested in seeing the details for that, I will direct you to our newsletter where the original solicitation is. That's going to be at our website at biogeography.org. You can go to this link, newsletter SU26, or you can find it through our link tree on the website. And on the topic of newsletters, the autumn edition will be coming out on October 1st. So, as always, as we get closer to that, I put out this solicitation for any kind of opportunities in biogeography that you want to share with the community. So, if you're hiring new students or postdocs or professor positions, if you have a cool workshop you want to share with everyone, something like that that you think people in this community would really enjoy and benefit from, you are very welcome to use our newsletter as a way to advertise that. All you have to do for that is to send the details to us at this email, biogeography at or .ibs at gmail.com. And as an aside, I would also say that this isn't restricted to our newsletter. So at any time of the year, if you have something you want to share, you can do the same thing. Email us, and we can send it on our website and our social medias at Facebook and Blue Sky. So let us help you advertise, is all I'm saying. As far as announcements go, that's all I've got though. So as far as actual seminar logistics, as always, this will be about a 45-minute talk from our speaker and then 10 to 15 minutes at the end for Q&A. If you have a question you want to ask, I'll direct you to the Q&A button at the bottom of your screen. You can click on that at any point during the talk and just type in your question there. At the very end, we'll have Marta read off the questions for you. So that way it kind of streamlines everything, but you can submit those at any time. You don't have to wait until the very end. And just a peek at our future speakers. So we have for September, Dr. Fang Liang He from the University of Alberta. That'll be at 1 p.m. UTC on September 30th. So details for that will come out once I have them. Keep an eye out for that as well. And as always, thank you so much for coming. If you want to see this again or you weren't able to come the first time around, these will be uploaded to our YouTube channel at International Biogeography. If you want to become a member of the society, you want to make a donation, or just keep an eye on what we're doing, you can check us out on our website at biogeography.org or our social medias. We have a Facebook page and a Blue Sky you can check out. And if you're interested in biogeographical research, then I would also direct you to our Frontiers of Biogeography open access journal that can be accessed at biogeography.pensoft.net. Lots of cool research that happens there. And that should be all from me. So I will pass it on to Marta to introduce our speaker. Thank you.
Thank you very much, Hikaru. Welcome, everyone. Thank you for joining us for our August Funk seminar. I am Marta Jarzyna. I'm the Director for Public Affairs and Communications at the TIPS. And it is my great pleasure today to introduce Dr. Daniel Cadena as our speaker. Dr. Cadena is an evolutionary biologist and professor at Universidad de los Andes in Bogota, Colombia. He received his bachelor's degree in biology from that university and then a PhD from University of Missouri at St. Louis. He and his research group study how species evolve, adapt, and diversify, especially in the tropics. And much of the work Dr. Cadena does focuses on birds, but he is extending his taxonomic focus to other groups as well, like, for example, cave-dwelling fishes recently. His group uses many approaches, including fieldwork, genomics, museum collections, ecological modeling, to explore the origins of biodiversity and how it is shaped by geography, climate, and evolutionary history. Over the years, his group has published on topics ranging from natural history, adaptation, speciation, to various aspects of biogeography, the ecology and evolution of animal migration, and biodiversity conservation. One of his main projects right now is a national biodiversity survey that retraces the paths of early naturalists to understand how Colombia's birds have responded to a century of environmental and cultural change. Dr. Cadena wears many hats. In addition to research, he is also a dean of the School of Sciences at his university. He is a president-elect for the American Ornithological Society, and he also does a lot of outreach in his native Colombia. If you go to his website, you can find some really fun outreach videos. So I encourage you to do that if you're interested. So without further ado, Daniel, welcome, and the floor is yours. Feel free to share your slides.
Thank you, Marta, Hikaru, and thanks everybody for tuning in. I'm gonna share my slides. Okay, can you confirm that?
Looks great.
Wonderful. Well, thanks again. It's a pleasure to be here. Good morning for those in much of America. Good afternoon to those in Europe. And I thought I would start today showing you this photo that I took in a recent trip to the high Andes of Peru, where I was amazed by the beautiful landscapes, but especially by the biodiversity in these places, which is very unique. So, like this polylepis tree at nearly 5,000 meters elevation, which caught my eye. But it's really amazing, like the variety of species that you can find such high elevations in these tropical mountains. And I was, of course, very interested in birds, like this finch, which we found at nearly 5,000 meters again on the shore of this very high altitude lake with this beautiful uh, snow peak around it. And I was really amazed by all the species that thrive in these ecosystems. And I thought I would start with this because mountains are global hotspots of biodiversity. And in a recent analysis that was just published a couple months ago or a month ago, led by Laura Schultz and Suzette Aflantua, we looked at biodiversity in alpine areas, like those I just showed you, like very high altitude, typically above the tree line. And we looked at the diversity of a bunch of different groups of animals and plants. And something I want to highlight from the results of that study is that we did not find the typical latitudinal gradient of biodiversity that many of you might be used to in other ecosystems. So yes, many tropical mountains are hotspots of alpine biodiversity, but there's no simple relationship between latitude and elevation. And one of the main conclusions of this paper was then that history probably matters a lot in terms of establishing global patterns of biodiversity. And that's one of the central themes of my research. And that's what I'll be talking to you about today, about how can we use evolutionary history to, tap into the mechanisms accounting for global patterns in biodiversity. So I typically work on mountains, hence the title of my talk. And this is one of the sites we work at, the Sierra Nevada de Santa Marta in northern Colombia, quite spectacular. And we're interested in the distributions of species along these elevational gradients and how diversity accumulates in mountains. And what I'll do today is sort of give you a flavor of the kind of work we do in these ecosystems. First off, I want to say that my work is highly collaborative. So I first want to acknowledge a lot of people that are co-authors and esteemed colleagues in the work I'll be showing you today. And also acknowledge that a lot of the work I've been doing over the years has been done with students, graduate and undergrads at Universidad de los Andes in Bogotá. So this is a photo from last month with showing a few of my students that we got together at a nice meeting. And a lot of the work I'll be showing you today has been done and inspired by them. Okay, so we all know that biodiversity is not homogeneously distributed. And to this crowd, I don't need to convince you that the tropics, and here focusing on the American tropics, is a hotbed for biodiversity. So on the left, you see the diversity of birds globally with a focus on the Western Hemisphere. So you can see a lot of red in lowland areas of and foothill areas of Northern South America. And the plot to the right, the graph to the right shows the distribution of restricted range species, those with small ranges. And you can see a pattern there that those tend to accumulate in mountains. So I live in Bogota, Colombia, in the Eastern Andes of Northern South America. So I'd say I'm in a unique setting to try to understand what and make sense of these patterns of mega diversity in this area. But just to give you a flavor of what mega diversity means, Colombia, as you might have heard, is the country of birds. We have close to 2,000 species in Colombia, of roughly 10,000 that occur globally. And just to give you a flavor for it, I put together a few slides that I hope you will enjoy. So when you think about Colombian birds, I just want to give you a sense of its staggering diversity by showing one kind of bird that some of you might be familiar with, a hummingbird, the hummingbird. But I like to say that when you think about Colombia and you talk about the hummingbird, that doesn't really make sense because we have lots of hummingbirds. And I'm going to show you a few slides of hummingbird species occurring in the mountain region of Colombia. And you can see, well, not only the diversity of species, but the diversity of interactions they have with other organisms, the diversity of their colors, of their behaviors, of the habitats they occupy, et cetera, et cetera. So diversity is multi-dimensional. It's not only a lot of species, but it's also a huge variety in different dimensions of such diversity. So none of those photos were taken by me. They were taken by Murray Cooper. You can look at the book on birds in Colombia we did with him a few years ago if you want to see more of these. And the other point I want to make is that diversity is not only very high in, I wouldn't say Colombia, but Northern South America, but is also still insufficiently known. So I've been fortunate enough to over the years, work with various colleagues in describing a bunch of new species of birds from this area of the world. I'm showing you examples of those here. So this is to make the point that not only is diversity very high here, but it's also insufficiently known. And of course, I care about this a whole lot. And the main question I'm interested in is why is diversity so high? And my research program sort of focuses on two main areas I want to explore. One is the question of diversification, and I'll talk about today about some old and some newer ideas on the origins of biodiversity. And then the other topic I want to touch on is to what extent are we conserving the ecological and evolutionary processes that sustain that biodiversity into the future? And as Marta said, we've been working on this re-expedition work that is using biodiversity surveys as windows to look across time. So I'm sorry if many of you came to this talk today hoping to hear a lot about that. I decided at the end, in the interest of time, I wouldn't talk about this a whole lot, but I will touch on it at the end. So please bear with me. I'll be happy to talk more about it later on. Okay, so the main pattern I want to focus on today is something that is probably familiar for many of you, is this figure that was first published by Alexander von Humboldt of the Chimborazo volcano in Ecuador, which shows how tropical mountain species replace each other with elevation. So Humboldt noted that the species that occur at lower elevations in the Chimborazo are not the same that occur at higher elevations. And this is something I'm very interested in, and I'm going to focus on that a little bit today. So a question I'm really interested in is the question of how do elevational replacements arise. If you take, say, two mountains shown here, where you have two pairs of species that replace each other with elevation, there are two sort of processes that could account for the origins of this pattern. One is what you would call a gradient model, in which the two species replacing each other with elevation in a given mountain descend from a single common ancestor. And then the other two species occurring in a separate mountain are in turn each other's closest relatives. And then the other model would be sort of the standard allopatric speciation vicariant model, where actually sister species do not occur within the same mountain, but occur on different mountains, typically at the same elevations. So this idea, of course, is not mine. This is something that was developed by Jim Patton and Mary Smith in a landmark evolution paper published in 1992 that has become sort of a framework for a lot of the work that we do. So why is this important? Why is it important to understand how do elevational replacements arise if we're interested in understanding global patterns in biodiversity? So first off, I'd like to show with this figure that mountains are central to understanding global biodiversity patterns. So this is work that was led by two of my former students, Elkin Tenorio and Paola Montoya. And what they're showing here is the latitudinal gradient, so latitude on the x-axis and then species richness in the y-axis for vertebrates occurring in both mountains and lowlands. So you can see that for both mountains and lowlands, diversity declines as you move away from the equator, so as you increase latitude. But I also want to note that the slope of this relationship is much steeper for mountain species than for lowland species. So the point being here is that mountains contribute disproportionately to the global biodiversity gradient. And of course, a lot of people have looked at the contributions of mountains to biodiversity. So this is work by Jan Fielsa and his colleagues showing that when you look at the richness of old and young species globally, shown by white in this figure, the tropical Andes and other mountain regions, such as the Himalayas or mountains in East Africa, emerge as places where both you get a concentration of old species, of species of ancient origin, and more recently derived species. And also, when you look at a very important component of diversity, which is beta diversity, meaning the turnover in species with in space, you find that the mountains also emerge as global hotspots, especially tropical mountains like the Andes, the mountains in East Africa, and the Himalayas are places where you move from one place to the other, you get a lot of change in the identity of species. And I want to argue today that that is a central component of the latitudinal biodiversity gradient and why tropical mountains are so important to understand such gradient. So what I'm showing you here are the elevational ranges of birds in three different avian families. So what I did here was tabulate their elevational ranges and stack them up. And what you can see here in the plots above is that for both flycatchers, ovenbirds, and tanagers, there's essentially no species that occupies the full range of elevation, say from sea level up to more than 4,000 meters. Instead, species have narrow elevational ranges and replace each other with elevation. Typically, congeners, for example, you might find one that occurs at low elevations and then it's replaced by a congener at higher elevations. So what we did in the plot below was to sort of summarize that data with histograms and box plots. And it's striking to me that across all these avian families, the median elevational ranges of species are about 1,100 meters. So these mountains, like I showed you at the beginning, can be really tall, more than 4,000 meters tall, but the typical species only ranges over roughly 1,000 meters elevation. So why does this happen is a central question in biogeography research in this region, and it's something I'm very interested in. And of course, this not only occurs in birds, but it also occurs in other organisms. So this is work that we did led by my former student, Paulo Pulgarin, looking not only at birds, but looking at parasites that infect birds. And here I'm not showing data from Colombia, I'm showing data from the other side of the world, from New Guinea. And here I'm showing you the elevational ranges of malaria parasites that infect birds. And you can see that also those parasites show this pattern of elevational replacement of closely related species. So I would argue that understanding the causes of these patterns is central to understanding the global biodiversity gradient. So one of the groups I've sort of tackled these sort of questions with is the Tapaculos in the genus Psilolopus. So it's a very complicated group taxonomically. Believe me, these 8 species shown here are 8 different species occurring on the same mountains of Ecuador. They're very hard to tell apart. But what you can see in the map to the right is the richness patterns in these organisms. So blue indicates old lineages in the family Rhinocryptidae, whereas green shades indicates species of more recent origin. And then the hue indicates a diversity. So you can see the Andes very clearly as places where you have one very high richness, and that is richness of species originating relatively recently. So we've become very interested in understanding how speciation proceeds in this system. And now I want to draw your attention to the plot on the right, and perhaps look at the boxes shown in orange and blue, corresponding first to the Sierra Nevada de Santa Marta, letter A, and the western slope of the Andes, the Cordillera Occidental of Colombia, letter B. And what I'm showing for those two places is that we have the typical pattern of species replacing each other with elevation. So two endemic species to the Sierra Nevada Santa Marta replace each other with elevation. This is an isolated mountain. So because these birds are very poor flyers, this is why I say they're mouse-like birds, one hypothesis we had is that maybe this isolated mountain was reached only once, and then you maybe had this pattern of parapatric ecological divergence, this gradient model described by Patton and Smith that I showed at the beginning as an explanation for elevation replacements. And then the case shown in B, it's amazing to me because you can find 6 different species in the same genus which replace each other with elevation. So again, we thought, well, it's highly unlikely that these birds that are very poor flyers would each come from different places to assemble along this elevational range. So maybe this would be a place where we could find evidence for this model of gradient divergence. So to test that, it took us many years, but we finally came up with a phylogenetic framework for the group in which we produced a high quality molecular phylogeny for the genus. And we simply wanted to ask the question whether species that replace each other on the same mountain are each other's closest relatives, which would support a gradient model, whereas the alternative case in which elevation replacements are not sister to each other, and instead sister species occur in different mountains, would support a bicariant allopatric speciation model. So after a lot of work, I'm summarizing that here with this figure. So again, let's draw your attention to the two mountains I showed before, the Sierra Nevada de Santa Marta and the western slope of the Colombian Andes. And what you can see with the phylogenies in the middle is that, well, it turns out that species on the same mountain are not each other's closest relatives. They, in both cases, are in different areas of the phylogenetic tree of the Tapaculos, which implies that even in these very poor flyers, species that replace each other with elevation did not evolve in situ from a common ancestor, but instead probably evolved in allopatry and came together via secondary contact. So we've done this sort of study in a variety of different groups, and what I just showed you, I think it's very general. And I think that an interesting framework to sort of deepen on our understanding of evolutionary mechanisms involved in speciation in these mountains is, of course, Dan Jansen's classic 1967 paper, which many of you are familiar with, I'm sure, which shows a central difference between tropical mountains shown above and temperate zone mountains shown below. So the plot shows, this is by the paper by Cam Galambor, which extended Jansen's hypotheses to an evolutionary mechanisms perspective. And what you can see here is that across the year in the x-axis, and you have temperature in the y-axis, tropical mountains show very little seasonal change in temperature. Consistently, low altitude sites are hot and high altitude sites are cooler. And Jansen's idea was that this should select for species with narrow physiological tolerances. If that is the case, then this would lead to great costs to disperse over a climatic gradient, be it crossing mountains from like crossing a high ridge or crossing a low-lying valley. And this would lead to low gene flow and eventually high allopatric speciation. That's what Galambor et al. Proposed. Alternatively, in temperate zone mountains, you got climatic seasonality across a year. So yes, typically lower altitudes are warmer than higher altitudes, but of course you have the seasonal component. So species that live either at low or high altitude in temperate zone mountains need to have broad physiological tolerances to deal with seasonality. So then the expectation is that those species would experience reduced costs to disperse over climatic gradients, which would in turn lead to high gene flow among populations and hence lower opportunities for allopatric speciation. So we sort of tested whether this would map to how do species form in tropical versus temperate regions a few years ago. And so we did an analysis that moved beyond birds, and we looked at a bunch of different vertebrates shown here. And what I'm plotting here, you can perhaps concentrate on the plot to the left, which shows the sum of the data for all the groups. And what I'm showing you is the degree of overlap in temperature across the year for the niches of species occurring in tropical mountains and species occurring in temperate zone mountains. So the main result here is that vertebrates in tropical mountains, sister species of tropical vertebrates, tend to have greater overlap in their thermal niches than sister species in the temperate zone. So what this says to us is that the niches of species at speciation in the tropics, at least in the tropical mountains, are highly conserved, which would be incompatible with a model of divergence across gradients, which I talked about before, but would be very consistent with a pattern where species originate within the same elevations in different areas. So again, allopatric speciation within thermal zones. And we think this is fairly general across animals, at least in the groups we have looked at. But of course, animals are not the only organisms we find in these mountains. So I started off talking about plants. So for this framework, thinking about plants is very important. So I told you at the beginning about Humboldt's work showing the elevational ranges of plants in the Chimborazo. Many of you might not know that a Colombian naturalist, Francisco José de Caldas, produced at essentially the same time as Humboldt very detailed profiles like those shown here of the elevational ranges of plants in the tropical Andes. So something we've been doing of late is trying to see whether the same speciation mechanisms that we found in animals so far also are found in plants, where again, in animals, we're finding typically allopatric divergence within the same elevation and not speciation across elevation within mountains. So what do we see in plants? So to get at this, I teamed up with a bunch of botanist friends led by Felipe Zapata and Adriana Sanchez. And we looked at sort of the same question we looked at with the animals. And here I'm showing you the same thing, the degree of temperature overlap between sister species of plants occurring in the tropical zone, in tropical mountains, and species occurring in temperate mountains. And again, we see that in the tropics, the overlap in temperature in thermal niches between sister species is very high and higher than in the temperate zone. So again, this suggests that speciation in tropical mountains, also in plants, is occurring largely within thermal zones. So I told you before that in animals, our inference was that, well, this probably means then that speciation is occurring in allopatry, because these animals we know typically occur in different mountains at the same elevations, but in different mountains. But there's an interesting twist to the case with plants, and I just got this from Felipe Zapata yesterday, where he looked at a subset of the organisms that we studied, so looking only at sister species of plants that occur in the same geographical area. So he did this by by looking at pairs that are unique to the same latitudinal band. So essentially, we're looking here at pairs of species that occur in symmetry in the same areas, and we're asking, like in regional symmetry, and we're asking whether those species occur in geographical overlap, which would mean in actual symmetry, or in little overlap, which would indicate allopatry. And the point here is that it's very interesting that tropical species of plants, sister species of tropical plants and mountains, shown in green, tend to have greater geographical range overlap than sister species of plants occurring both in the north temperate zone and the south temperate zone in mountains, which means that maybe there's something different going on in plants. So maybe we do have speciation within a thermal zone, which was shown by the earlier plot I showed you, but in contrast to what we see in animals, we don't rule out the possibility that in plants we might have more of what one might call sympatric speciation. So at the within a thermal zone in the same region, perhaps via other mechanisms than those we know occur in animals. So of course, plants do funny things such as hybridization, such as changes in ploidy number, those mechanisms that might promote in situ speciation. So we're keen to explore this a little more because we think it's very interesting. Okay, so to continue talking about this Janssen framework, which I think is very interesting, something we want to do is so in general, this Janssen's idea that there is little overlap in thermal regimes between low and high elevations in the tropics and greater overlap in the temperate zone, that sort of makes sense in general. But the relationship between elevation, or say the distance in elevation between two sites, and the degree of thermal overlap between those sites is not the same depending on things like the shape of mountains, the size of mountains, whether they face the ocean, whether they face forests, etc. So we've started doing things like this. So this is work we're doing with Ignacio Quintero, Juan Parra, and Ethan Link. So if you look first at the figure to the left and look at all these red dashes, so those are our transects that we use GIS to put them on a map. And along those transects, we recorded the distance in elevation between pairs of points. So that's going to be plotted in the x-axis and the plot to the right. And the degree of thermal overlap between those points shown in the vertical axis. So what this plot shows is that, sure, the greater the distance in elevation between any given two sites in this mountain shown here, the lower the degree of thermal overlap. So climates are more different between sites that are farther apart in elevation, but you can see that the slopes of change are very different. So the degree of overlap between low and high elevations in different mountains depends on the mountain. So if that makes sense, what I'd like to do now is show you how does this look when you take the same sort of analysis and do it globally, looking at many different mountain systems all over the world. And what you can see here is that there are very interesting patterns where I just show you the data for the Andes, but if you look at, say, mountains like those in New Guinea, another typical tropical mountain system, you see very little overlap, regardless of which mountain you are located at, between low and high elevations. But then if you go to other places, like maybe mountains in Central America or other places, the degree of overlap between low and high elevations in thermal regimes might be greater. And also, it's very interesting that there are some mountains in the temperate zone that show very little overlap in thermal regimes between low and high altitude. So the reason why I'm showing you this is that we think that Janssen's hypothesis is a powerful explanation for patterns in global biodiversity, but that it could use some refinement if we actually quantify the degree of thermal overlap across elevational ranges, especially if we map that in relation to variables of interest for those evolutionary biologists and biogeographers who study this sort of things. So for example, Ignacio Quintero and Walter Yates published this paper in Nature where they looked at how do species richness and diversification rates vary with elevation in different mountains of the world. So what we're hoping to do now is sort of match our earlier plots with this sort of plots and see whether we can get better explanations for these other patterns. Okay, so as like Marta mentioned before, I've worked mostly on birds, but I just showed you with data from plants, I've moved into other systems. But a system I'm increasingly interested in because I still think that despite not having found good evidence for gradient speciation in birds, I think it's very plausible in mountains, in animals, in the tropics. So instead of looking at elevation gradients, we've started looking at depth gradients. And it's remarkable because in the eastern Andes of Colombia, we have this karst cave system in which we find these magnificent creatures. So this fish above is Trichomicterus rosa blanca, endemic to the eastern Andes of Colombia, which it's, well, remarkable in many ways but it shows the typical troglomorphic phenotype that has evolved in a bunch of different animals that have adapted to life in cave systems. So you can see this a fish here lacks eyes completely, lacks pigmentation. And a phylogenetic study we did revealed that its closest relative is apparently Eremophilus mutisi, which interestingly was described by Humboldt, the fish below, which is very dark and pigmented and has eyes and lives in surface rivers here in the Cordillera Oriental of Colombia. So we're actively studying this system, and what I can tell you now, based on a phylogenetic analysis we published a couple of years ago, is that we're very excited because I can tell you all the details here, but I just summarize by saying that we found at least 3 and likely 4 independent events of colonizations of caves from surface ancestors. And in all those cases, we see sort of trends in the same direction of losing eyesight, losing pigmentation. And in some of those cases, we're seeing the possibility that these events might be taking place in the phase of gene flow without any obvious barrier to the movement of genes between surface streams and cave streams, implying that adaptation, natural selection to different conditions might be promoting the origin of biodiversity in the system. So in a sense, I'm not discarding entirely the possibility of a grading model of speciation in animals in tropical mountains, only that we've been perhaps looking at the wrong organisms in the wrong environments. We should have been looking at fishes in caves instead. So if you're interested in this, keep an eye out as we'll be working on this a fair bit over the next few years. Okay, and the final point I want to make about this topic about elevational replacements is that I think a very important component here is the role of range dynamics of expansions, contractions, and extinctions in explaining patterns of elevational replacements with elevation. So this is an example of a study we did with Henicurrhina woodwrens a few years ago. So these are patterns of genetic structure within this species complex. And what I did here was plot lines that connect genotypes to locations on the map. And what you can see here is that's pretty amazing to me that all these lines sort of cross each other a whole lot. So 2 things I can tell you about. So for example, shown in blue, Encurrina negreti, endemic to the western Andes of Colombia. Its closest relatives are not in the mountains of Colombia, but instead seem to occur in Middle America, in places like Guatemala. And then another interesting case are populations from northern Venezuela seem to be most closely related to populations from Bolivia than to nearby populations from the Andes of Colombia. So the point here is that I don't think that these little birds probably flew from, say, Guatemala to Western Colombia or from Bolivia to Venezuela to establish new populations there. But probably what this reflects is that probably these species have gone through phases where the ranges expand and they colonize new regions, then they contract, you get local extinctions, and then you get this sort of mosaic where you have these distant populations that are closely related to each other. So range dynamics, I think, is a very important factor in accounting for these patterns. So that said, then how does secondary contact arise? Why would species go through phases where they expand and then contract? And when they expand and get isolated, they might diverge to form new species, which then clump together in secondary contact to contribute to diversity in mountains. So a hypothesis that makes a lot of sense is that climate fluctuates through time. So this is, of course, nice work by my colleague Suzette Flantois, looking at how climate has influenced the connectivity of high Andean vegetation in the northern Andes, and specifically the páramos of Colombia, throughout the Pleistocene, where you get these cycles of climate, which have presumably led to expansions and contractions of forests. So a hypothesis we might entertain is that those expansions and contractions lead to isolations of populations, and then during periods of climate change, these populations might come together, leading to the buildup of symmetry in mountains. So to address that, something we can tap into is information contained in the genomes of species occurring in the Andes. So specifically, if we can reconstruct demographic history showing eventual changes in population size through time, a pattern where you see marked fluctuations in population size, which would reflect population growth where environments expand, and then population declines when populations where environments contract. This would perhaps be evidence of the influence of these climatic cycles on these cycles of species range dynamics. So we just published a paper on this, looking at a hummingbird genome, a hummingbird species from the Colombian Andes. So we sequenced a reference genome for this species, Eleangelus exortis, and we did resequencing for about 10 individual animals. And here what I'm showing you is effective population size through time, and you actually see this pattern of apparent range dynamics coupled to changes in population size, where populations initially increased in size, then declined towards the present. And this perhaps could be consistent with a model of range expansions and contractions driven by climate change. So we're now trying to expand this to other species. And so far, it's very interesting because we see a bunch of different patterns. So what we did here was for all these species shown here from the very same site, we sequenced whole genomes for a bunch of individuals, reconstructed demographic history. And you can see that there are some common patterns where species show sort of concurrent patterns of population growth and decline, but others show very idiosyncratic patterns. So what we're hoping to do is sort of assemble more data here and perhaps get a greater, more comprehensive picture of how things change over time. And of course, we have seen this in other systems, and this is work by my former student, Maria Isabel Castaño. She looked at sort of the same sort of thing. So the x-axis shows population size, time, sorry, and the y-axis shows population size through time. So here she's looking at two different species, and you can see the signature where first you had a population decline, where species likely were isolated in refugia, and then a population expansion. And I'm showing you these two together because these two species are species that are, or populations that are co-distributed, so they occur in the same mountains, but they show these sort of range dynamics that suggest the possibility of populations being isolated at some point in the past and then coming together. And of course, we know that they come together because they meet each other along elevational gradients in the western side of Colombia that Maria Isabel has studied in great detail, where they hybridize. So what I want to say here is that not all those events of population divergence and subsequent meeting and secondary contact lead to diversity accumulation, because sometimes speciation has not proceeded to the point of you having reproductively isolated species, and that you get sort of hybridization and the erosion of diversity. So if you want to read more about this, Maria Isabel just published a very nice paper in Evolution looking at the genetics of these hybrid zones and actually getting towards pinpointing the origins, the genetic basis of phenotypes in the system, which is another dimension of biodiversity that we're very interested in. And then another example we've been working on are these warblers in the genus Myoboris, which you can see here, two forms that meet in southern Colombia and northern Ecuador. And in this case, we also see hybridization in southern Colombia and northern Ecuador that leads to these very interesting pattern where you get all these mixed phenotypes in the hybrid zone. We also just published a couple papers led by former student Laura Céspedes in the latest issue of Evolution. But the reason why I'm mentioning this case in particular is that we published this analysis or this perspective a couple years ago in one of the society's journals, Frontiers of Biogeography, which I actually like a lot. This was led by my colleague, Maria Jose Sanin. And one of the things we argued here is that we typically think about species changing their ranges because of climate change that I showed you before. But something we argued here is that volcanism, so the eruptions of volcanoes that generate topography might actually connect populations that were formerly isolated. And this would lead for these opportunities where you could either have species colonize areas where they weren't before and then contribute to local diversity, such as the case of wax palms that Maria Jose has studied, or in the case of the Werber hybrid zone that we've studied, led by Laura, this leads to hybridization and perhaps the erosion of incipient speciation. Okay, so I'm getting to the end now, and I promised I would say at least a few words about the second question here about whether we are conserving the ecological and evolutionary processes that sustain biodiversity. So I won't talk about the resurvey work today, but what I'll do is sort of talk about sort of the implications of what I've told you about so far. So to summarize, what a main take-home message from the talk today is that we think that a lot of speciation in tropical mountains proceeds by a allopatric model, a vicariant model, where populations occurring in different mountains at the same elevations are each other's closest relatives. So we showed that in the Tapaculo case, but we also published this paper a few years ago with colleagues Ethan Link, Ben Freeman, and Cameron Gallambor. And this seems to be very general for at least mountain birds in tropical areas in different parts of the world, including Africa, New Guinea, the Himalayas, etc. So based on that, what we think then is that through evolutionary time, species tend to conserve their ecological niches, especially as it relates to temperature. You typically don't see species from tropical mountains that occur like closely related species that replace each other with elevation, implying that divergence at speciation was caused by niche divergence. Instead, we think that niche conservatism is what isolates them in separate mountains at the same elevations. And this idea that niches are conserved at speciation, we think has important implications for conservation, thinking about things such as global change. So one of the things we argued in this paper is that the fact that thermal niches of mountain birds appear to be evolutionarily conserved, as shown by the kind of work I've showed you, implies that it's unlikely that they'll be able to respond to climate change via adaptation. Their thermal niches have been stable for millions of years, so it seems unlikely, at least intuitively, that they will be able to respond by evolving their thermal niches to adapt in place to things like global warming. So instead, we argued here, what species will need to do is to be able to move, to track those environments that are suitable for them, given their specialized thermal niches. And for that, we're going to need things such as high connectivity across elevations. So I think this works in general, but the final example I want to show you has to do with the fact that recent reviews looking at contemporary changes of bird populations to climate change are not uniform. So something we're seeing is that many species are indeed moving their elevational ranges to higher elevations, as you would expect given climate change. But in many species, we're seeing no changes. And even in some species, we're seeing downslope shifts in the elevational ranges. So something we decided to look at is whether we can use sort of the evolutionary thinking framework that I've proposed so far, where we use evolutionary information to perhaps inform about the potential responses of species to contemporary global change. So this was work that was done by Jessica Urbano as part of her master's, which she finished recently. And what Jessica did was, in this first analysis, she took a global phylogeny of birds and mapped elevational ranges onto it. And the point here is that there are some lineages in which elevational ranges have indeed been very conserved, as implied by everything I've showed you so far. But in other cases, evolutionary changes in elevational ranges have been more common. So elevational niches have been more labile. So then the question we wanted to address was whether species that over their evolutionary history have had more labile niches have had different responses to contemporary global change than those species belonging to lineages where thermal niches have been highly conserved. So this is the final figure I will show you. So here Jessica got data for more than 200 species from tropical mountains for which we have data on changes in their elevational distributions over the past century. So that's shown in the vertical axis. And then for each of these species, she estimated their evolutionary rate in their thermal niche from the comparative phylogenetic analyses I just showed you. And I think the answer is very interesting. So first, it turns out that for species in which niches have been more labeled evolutionarily, so species to the right in this plot, they tend to have shifted less in elevation uh, over the past century. So I think this makes some sense because what this implies is that species that have more labile evolutionary niches might be more plastic and then do not need to move in response to global changes in temperature being caused by humans over the past few decades. But if you look at this figure carefully, you can actually see that the fit to the regular regression line is rather poor, but instead you see a figure that looks like a triangle. And you can see there's a white space in the upper right. So we don't see species with high rates of niche evolution that have shifted a lot in terms of elevational distribution. But when you look at highly conserved species, those have species belonging to niches to clades with highly conserved thermal niches, you see all sorts of responses. So some of them haven't changed their elevational distributions at all. So we don't know if these are on the way to extinction or they're being able to respond in situ, but a lot of them have had to move and moved a lot. So I think this only reinforces our point that in order to conserve these evolutionary processes into the future, we're going to need to maintain connectivity across mountain slopes. So with that, I'll end just by saying that another take home, I think, from all this talk is that looking at these patterns that have inspired naturalists since Caldas from an evolutionary lens leads to insights that I hope you might have found of interest today. So with that, I'll end thanking a bunch of funding sources and thank you all again for attending. I'll be happy to take questions if we have time, Marta.
Thank you, Daniel. This was wonderful. We have several minutes for questions, and there are already a couple of questions that I will read to you. The first one comes from Adela Roa Varon. What molecular data were used to construct the phylogenetic relationships presented?
That's a good point. So I've been for a sufficient time in this field that the paradigm has shifted. So when I first started working on the phylogeny of the Tapaculos, essentially what we did was sequence mitochondrial DNA. And it took us so long. So here's a message for students: don't sit on your projects too long so as to so that the paradigm changes. So the point when we were ready to publish the phylogeny, they wouldn't publish that phylogeny based only on mitochondrial DNA. So we went and teamed up with colleagues who were able to get genomic data using UCE data. Sequences flanking ultraconserved elements. So that's what we use mostly. The good news is that the trees using mitochondrial DNA and the genomic data are essentially the same. So a lot of what we did earlier on was based on mtDNA, but we've moved on to using things like RAD-seq, low coverage whole genomes. That's what we're doing now.
Okay, great. Thank you. Next question comes from Mark Lamolino. Intriguing insights, Daniel. In some mountain systems, we see shifts in ranges in apparent response to the presence or absence of putative competitors. To what degree are the patterns you see influenced by species interactions?
That's a great question. I didn't talk about, and actually that was the topic I worked on for my PhD many years ago. And I think that interactions are very important. So I mentioned at some point that some species have been able to move up to higher elevations and some haven't responded. So one of the reasons might be because there's a congener up there that is a better competitor at higher elevations. So maybe species are able to track their changes in climate and move upslope, but interactions might impose limits into what might be possible. But then the other interesting thing is that maybe with all these disruptions in climate, which not only involve changes in temperature, but also precipitation and other variables, the dynamics of those interspecific interactions might change. So we might live in a situation where you have, stable range boundaries set by, say, competition. But what if you shuffle the environment? So a given species might be a better competitor at higher elevations given the current temperature and precipitation regimes. But if you change those two things, it's kind of unpredictable what might happen. So species from other elevations might be able to move in. So I think it's a very interesting topic, but it's a lot harder to study.
Okay, great. Question from Stanislav Walasek. Thank you very much for interesting presentation. I have two questions. The first one is if you did find any evidence of changes in differences in pigmentation in studied bird genera or families, which could confirm or indicate validity of the Bergmann's rule in this question. And the second question is if inside the same studied region, studied taxon groups, apparent any impacts of altitude changes to the frequency of sexual dichromatism? Both of the questions are connected to the context of limitation of sources connected generally with increasing altitude.
Yeah, that's a very good question. We haven't investigated any either of them. Um, there is some evidence in DM birds. This is not work by our group, showing that we do see patterns consistent with Glover's rule in some groups. So you get darker birds with more melanin in more humid places, and the idea there is that melanin might confer um, protection against bacterial growth on feathers. So there's some evidence of that. Um, and then, yeah, the question about dichromatism, so species in which perhaps there's greater sexual selection responding differently. I think that's an interesting idea as well, but I haven't looked into it, and I wouldn't have like a clear intuition of what to expect. It'd be interesting to look at.
Just another thing to add to your portfolio. Question from Richard Field. On the final research you showed, it was absolute elevational shift. But did you look at differences between upward and downward elevational shifts? And why do you think some species move downward?
Yeah, that's a great question. Actually, Jessica, I showed you one of the plots, but she did like a bunch of different things. She looked at the shift in the median elevation, the shift in the upper limit, uh, and actually, like you said, some species move upwards and some moved downwards. We plotted the absolute shift just to make it simple, but we do see things moving downwards in some cases. And then I think interactions is one possibility. So, like I said before, but then the other thing is that these mountains systems are faced to a bunch of different threats at the same time. So it's not only climate change, but you might have things such as deforestation. So sometimes, the perhaps more immediate effect of deforestation is to push things down, especially if you have greater deforestation high above. Whereas under climate change, you would expect things to move upwards. So it's hard to tell exactly what's going on in a case where we haven't actually followed the individual species through time to see what's happening.
Yeah, it's certainly a complicated issue, and we know that land cover change and climate change interact with one another. So that's something that we need to be looking at. Well, thank you very much, Daniel. It was wonderful. We got a bunch of comments in the chat as well, saying how interesting the talk was. Oh, there was actually one other question that we have 1 minute to maybe answer.
Uh-huh.
Um, okay, let me decipher this. This is from Abdul Samad. How do we prevent the current shifts of not only animals but also plant seeds, spores, and soil? Can you like give us some advice? Is I think what Abdul is getting at. Like, can we?
Yeah. Actually, I'm not sure Abdul, if I understand the question correctly, but I would say we shouldn't prevent the current shift. Actually, range shifts might be the only way that species are going to have to persist in these modified landscapes. So, if anything, we should encourage having landscapes that allow species, if they need to do so, to move across elevational ranges. So, rather than prevent it, I think we should foster it.
Excellent. This is a great point to end on. Thank you again very much, Daniel. Thank you all for coming. We had a wonderful attendance of over 130 people. Fabulous. If you want to rewatch the talk, it will be on our YouTube channel. Send it to your colleagues, send it to your friends, and join us for the next installment in September. Thank you again and have a great rest of your Wednesday. Thank you, Daniel.
Thank you all. Have a good day.
Bye, everyone.